Hi, everyone. My name is Shruti Abbato, and I head up business development at CARMA Cell Therapies. Thanks for your interest today. CARMA is a clinical stage cancer cell therapy company founded by MXCT in 2015. Our core focus is on mRNA-based immune cell therapies, which are a largely untapped class of targeted immune cell therapies for treatment of solid tumors. Our disclaimer. CARMA is MXCT's wholly owned subsidiary. MXCT is a publicly traded company developing flow electroporation technology to enable non-viral cell engineering applications. This MXCT technology is the subject of multiple high-value agreements with leading cell and gene therapy companies. MXCT conceived of CARMA's mRNA cell therapy platform as a proprietary application of flow electroporation to solve safety, efficacy, and manufacturing limitations of conventional CAR T cell therapies. MXCT has since invested over $34 million to build CARMA as a clinical stage company and is now seeking $50 million in independent funding to accelerate CARMA's clinical and preclinical pipeline. CARMA operates independently as MXCT's subsidiary with a dedicated organization. We have a strong research team, a strong clinical team, all guided by highly experienced cell therapy, clinical, and regulatory experts and leaders. We've established GMP manufacturing for our cell therapies through in-house technical development and our CDMO. And with MXCT, we share our senior executive leadership and G&A operations. CARMA's mission is to harness our mRNA cell therapy platform for developing autologous mRNA cell therapies to address limitations of conventional CAR T therapies. Our mRNA platform is non-viral. It uses unmanipulated multi-effector immune cells. Its cell modification is transient as a result of mRNA, and it allows for rapid one-day autologous manufacturing. Harnessing these critical platform attributes, CARMA is developing a pipeline of first-in-class mRNA cell therapies directed to highly attractive solid tumor targets to drive greater efficacy, safety, and manufacturing simplicity. In our platform, PVMCs, instead of a single cell type, are isolated from patients' apheresis with no further ex vivo selection or chemical activation. Then using MXCT's non-viral flow electroporation technology, these PVMCs are transfected with one or more mRNA for the CAR with no ex vivo expansion. Under this one-day process with limited ex vivo cell manipulations, the result is a multi-effector CAR PVMC cell therapy comprised of critical innate and adaptive immune cells, all transiently expressing the CAR and also retaining a desirable naive cell phenotype. We're starting to understand the potential killing mechanism of our multi-effector CAR cells. We know our transient CAR PVMCs kill through direct CAR signaling. But because our CAR PVMCs have all critical immune cells, we have emerging data now showing that the CAR PVMCs also broaden and extend the tumor killing beyond adaptive CAR signaling. We believe this could be a direct result of the activated APCs in our CAR PVMCs, enhancing antigen presentation and facilitating epitope spreading. It may also result from activated B cells in our CAR PVMCs producing anti-tumor specific antibodies. This mechanistic work is ongoing and provides strong rationale for CAR PVMCs as a novel targeted immune cell therapy for solid tumors. Our platform, as a result of mRNA transfection, enables great flexibility in cell engineering. Our cell engineering is guided by three principal cell enhancement strategies to improve the antitumor efficacy of our mRNA cell therapies. by driving greater epitope spreading, by counteracting the tumor microenvironment, and by improving endogenous effector cell fitness. Guided by these three enhancement strategies, we will utilize a combination of proprietary targeting and modulator ideas identified by our research team to develop our mRNA cell therapy pipeline. Autologous manufacturing of our cell therapies is rapid and uses non-viral flow electroporation for transfection of mRNA into unmanipulated PVMCs. It avoids the complex and time-consuming ex vivo cell selection, chemical activation, expansion, and GVHD reduction-related gene editing steps associated with manufacturing of conventional autologous CAR T cell therapies. As a result, our manufacturing takes less than 1 day from receipt of apheresis to cryopreservation of the product. And our commercial goal is to truncate the vein-to-vein time for patients to less than 1 week. So the rapid manufacturing, the less than 1 week vein-to-vein time, plus the lack of expensive viral vectors, all provide significant cost advantages over conventional viral-based autologous cell therapies, and also have potential to be competitive with off-the-shelf allogeneic approaches. To summarize our platform, mRNA cell therapies, we believe, offer multiple advantages over conventional CAR Ts. The multi-effector population in our product leverages both adaptive and innate anti-tumor immunity. This may improve overcoming the immunosuppressive tumor microenvironment and also facilitate broader epitope spreading. The cells experience limited ex vivo manipulation and retain desirable naive phenotype, reducing cell exhaustion and improving tumor trafficking. mRNA-based transient CAR expression has potential to self-limit on-target off-tumor tox and avoids the safety risks in monitoring associated with viral vectors. mRNA also enables flexible cell engineering to express multiple molecules. And finally, our rapid autologous manufacturing process has potential for short vein-to-vein time and low cost of goods. We're building an innovative pipeline of mRNA cell therapies using our platform to treat solid tumors. The 50 million round that we're seeking will build a company with 4 products and achieve multiple value-creating milestones. Our lead program, MCYM11, is directed to high mesothelin expressing tumors. It is the clinical proof of principle for the CARMA platform. It's in a first-in-human phase 1 study as a single-agent intraperitoneal therapy in patients with advanced ovarian cancer and malignant peritoneal mesothelioma. We plan to expand MCYM11's phase 1 evaluation as a single-agent IP and IV therapy and advance it to a meaningful clinical signal by 2022. In addition, we plan to develop 3 new mRNA cell therapies. CARMA has spent significant resources to identify attractive solid tumor targets and also cell engineering strategies to enhance anti-tumor activity of our therapies. We're implementing these ideas to develop a second generation MCYM11 and 2 confidential programs, all to IND by early 2023. Mesothelin is an attractive solid tumor target. Its expression is highly elevated on multiple solid tumors with limited expression on normal tissues. Our first target indications for MCYM11 are advanced ovarian cancer and malignant peritoneal mesothelioma. Because both have high mesothelin expression, both are very difficult to treat, including with immunotherapies such as checkpoint inhibitors, and both have potential for regional intraperitoneal delivery. Currently, there are no approved therapies targeting mesothelin. A number of cell and antibody therapies are in development, with the most advanced in phase 1-2 development. So MCYM11 is neck and neck with other clinical candidates targeting mesothelioma. Our first in human phase 1 study with MCYM11 is a single agent 3+3 dose escalation study evaluating 4 dose levels. The patient population we're treating present with highly challenging refractory disease. Patients also don't receive preconditioning in this study, and in dose levels 1, 2, and 3 only received a single cycle of therapy. Despite this, interim results from the first 3 dose levels, which were presented this year at ASCO in a poster discussion presentation, showed promising safety and durable stable disease of up to 6 months in 5 of 9 patients reported. Enrollment is ongoing in the 4th dose level with expected completion later this year. Our development plan for MCYM11 seeks to create multiple options for its accelerated registration as single agent, intraperitoneal, and IV therapy. The current IP study is ongoing. We recently initiated a parallel IP cohort to allow for preconditioning, a step which is included in most CAR T treatments to improve response, and also to allow for multiple cycles of therapy given the transient CAR expression of our approach. Early next year, we will initiate a phase 1 IV basket study in patients with high mesothelin expressing solid tumors. This will be a lead-in study for phase 1 IV disease expansion cohorts. Compelling results from any of the phase 1 IP and IV studies could trigger registrational phase 2 studies in 1 or more mesothelin expressing solid tumors as early as 2022 within the funding horizon. In summary, CARMA is a platform company with its lead solid tumor program in the clinic. Our mRNA cell therapy platform offers efficacy, safety, and manufacturing advantages over conventional CAR Ts. We're building a novel pipeline of mRNA cell therapies directed to highly attractive solid tumor targets, with our lead clinical candidate showing promising preclinical efficacy and interim clinical safety and durable stable disease. We've established a strong scientific and technical team, strong leadership, and full business operations, as well as a broad IP position to move CARMA forward. Our financing goal of $50 million enables multiple clinical and IND milestones in the next 2 years to support future financing, partnership, or exit options for CARMA. I thank you for your time, and please reach out to any CARMA or MaxCyte colleague if you're interested in learning more.